So we are using the MxP Quant 500 kits from Biocrates for the absolute quantifications of metabolites from plasma samples. So the idea comes from that. Despite the importance of metabolomics in understanding of human health and disease, a comprehensive reference range for human plasma metabolites specific to the Indian populations remains elusive. Because the challenge is that the vast number of metabolites present in plasma poses a significant challenges for absolute quantifications in a single analytical rod. So in this study, we employed the MxP Quant 500 crate from Biocrates to quantify approximately 500 metabolites from human plasma samples in a single LC-MS run. Plasma samples, because this work is under a big mega project, CSIR cohort projects, which is called a Phenome India Project PI check. So under the aegis of Council of Scientific and Industrial Research. So this is the work package of that project. So this pioneering study lays the foundations for establishing a pan-India reference range for human plasma metabolites, facilitating the future metabolomic research and biomarker discovery, which can tailor to the Indian populations. So I will little discuss about the Pan-India project, the Phenome India projects. So in these projects, we're trying to develop a useful personalized risk prediction score for the cardiometabolic disorders, the non-communicable disease. So it is right that this personalized scores depends also the population based, because whatever we are saying the standard or reference range, it is totally the geographical specifics and the specifics for the habitants. So there is till now no database for the specifically for the Indian populations. So in this study, we are trying to collect the plasma samples from a pan-India basis. So this is a pan-India based sample collection. Till now, we have collected 10,000 samples, although the sample is not too much, but it represents the pan-India because we collected the samples, the different CSIR labs. So there are 37 CSIR labs and have some outreach centers throughout the India. So this is the locations of the CSIR lab. You can see some north, east, and west. We are divided in as per east, west, north, and south. So in this study, we collected the data, both the pathological and medical data and the molecular data. So the pathological data, we collected the ECGs, body composition analysis, anthropometric analysis, general blood chemistry, skin chemistry, liver function state, and lung function state is the spirometry, oscillometry, etc. And for the molecular study, we study for the RNA-seq, cytokines, microbiomes, GSA, microRNA, metabolomics, telomere assay, and also proteomics, metabolomics, and lipidomics. So today I'll discuss about the metabolomics study under this mega project. So in this metabolomics, we have a target to analyze the 5,000 samples. Plasma has samples from the different region of the India. So till now we have analyzed 1,000 samples, but quality check we have done and we have analyzed only 463 samples. So I'm going to the result part. So till now we have analyzed 469 samples. Among them 235 are males and 234 are females. So in this Biocrates kit, we are targeting 630 metabolites, including small molecules and lipids. There are 107 small molecules and lipids are 523. So in small molecule, we have studied the LC-MS method we adopted as per the standard protocol of the kit. And for lipids, we adopted the FIA-MS method, which is flow injection analysis. In this case, we are bypassing the LC part. We directly inject the samples into the mass spec. And so in small molecules, we have divided in different groups like alkaloid, amino oxides, amino acids, and bile acid, biogenic amines, carboxylic acids, fatty acids, hormones, cofactors, indoles and derivatives, nucleobases, vitamins, and carbohydrates. And in lipids part, we have the different class like acyl carnitine, lysophosphatidylcholine, phosphatidylcholine, sphingomyelin, ceramides, and there are different types of ceramides, cholesterol ester, diglycerides, and triglycerides. So if I'm not discussing the detailed workflow, just a snapshot. So in one plate, it will take around 50 to complete run. It will take around 58 to 60 hours. So we can divide it the workflow like a LCMS setup. It will take 4 hours. Sample PPA preparation 4 hours. LC plus FIA means the small molecules and the fatty and the lipids. The run it takes 48 hours. Data processing 2.5 hours to 3 hours. So one plate we can analyze 79 samples. For the 79 samples, it will take 60 hours to analyze approximate 500 metabolites. So this is the sample preparation protocol. I'm not taking too much time to. So in this plate, 996 plate, apart from the samples, there are blank, means zero sample, and there is also geosample. Geosamples means we use a PBS because this PBS has the same ion suppression effect and salt concentration as per the plasma. So here we use the zero sample as a plasma. And there is a calibration standard and also the quality control samples. Then we transfer the samples. Here we need only 10 microliter sample to analyze 500, more than 500 metabolites. And then we need some derivatizations for some fatty acid products, metabolites. So we have to derivatize some fatty acids. After that, we have eluted the samples in methanol, amino acid methanol in the actual plate. Then it centrifuge it, and then it will be subjected to the mass spectrometry analysis. So now the quantification strategy. So here in this, as for the kit and the protocols, we have different types of quantification strategy. Some are quantitative, so some metabolite from LC and FIA part will analyzed in a 7-point calibration mode. So this is called the quantitative. And some metabolites for the LC part will use the quantitative with restrictions. Means here we use only 1-point calibration. This is the standard protocol in the kit. And some are the relative quantitative. So in this case, because we are using the quantification in respect to standard, but in case of some metabolites, the actual standard is not available. So we are sharing the same chemical group standard. So it is called the relative quantifications. And unfortunately, the fat we experienced that the fatty acid 12-0, fatty acid 14-0, and fatty 16-0 and 18-0 is invalid because these fatty acid generally it is a contaminations comes from the solvent. So we cannot avoid that. So these fatty acids we cannot detect by this method, and it is also cited in the protocol. So now come to the result part. So first I'll show the what result we got, and then I will compare the data of the male and female, the same metabolites. So here in acyl carnitine, it is one type of lipids. So acyl carnitine, you see that the metabolites names are C0, C2, C3, C3DC, C14-1, C14-1. Different classes are there. This is the male concentration in median concentration of the 235 males. Is the female concentration of 234 females. And this is the reported reference range. So I'm saying here, this reported reference range is not for the Indian populations. We have collected from the literature survey in different locations, different habitat. These are the reference range. And it got, because our sample is a normal sample, and we got the range in somehow near about the reported reference range. And these are the amino acids. You also see that amino acid range is also similar. Our normal subject amino acid range in male and female is very close to the reported reference range. And these are some the amino acid related compounds like your betaine, creatinine, cysteines, homocysteines, kynurines, serposins. This is also complies at most of the within the reported reference range. Taurine. These are bile acids, and bile acid. But unfortunately, the bile acid is less in case of some of the. So bile acid we cannot compare it because the range is not as per the reference range. And some biogenic amines is also falls under the reference range. Carboxylic acids, whereas the aconic acid, hippuric acid, lactic acid, tetradecanoic acids, and also some of the carboxylic acid reference range is not available throughout the world. Like your aconic acids and tetradecanoic acid, there is no reference range. And crystals, the examples of fatty acid. Fatty acid is also about there is no reported reference range. Hormones, only cortisols which we got, and case of indole and derivatives is the 3-IAA, 3-IPA, and 3-endoxysulfates. And here also we did not find any reference range for these indole derivatives in human plasma samples. And vitamins and polys, the cofactors, it is also below the reference range what we got as compared to the literature. So this is because I'm saying that this is a preliminary study, so I cannot strongly say anything about because we had to do more analyze and more samples. This is only the presentations of only 469 samples. So if we compare in these small samples, there's some crucial metabolites between male and female expression. Here you see that most of the metabolites are significantly in case of acyl carnitines like C0, C2, C3, C14:1, C14:2. So most of the acyl carnitines in male, it is significantly high, right? And also trimethyl amine oxide is a biogenic amines. It is a choline derivative products. So it is also have no significant changes between male and females. But most of the amino acid, except cysteine and serine, all of the amino acid have significant overexpression in male compared to female in Indian populations. It has with the alanine, arginine, asparagine, aspartic acid, but not the cysteine. Also, lysine, leucine, isoleucine, histidine, not glycines, also methionine, phenylalanine, proline, not serine, because serine and cysteine have some similar structures. So these two amino acids have no significant expression changes in case of two sexes, male and female. But apart from that, other amino acids, all of the amino acids have showed significant expression changes in higher expression in male compared to female. And valine also showed the higher expression in male. And the bile acids, in case of only oleic acids, only CDCA and GCA showed the significant overexpression in male compared to female, but other like folic acid, DCA, GDCA, GLSA, GLCAS, there's no significant changes. And taurocolic acid have a changes, significant expression. Beta-alanine also have And in case of biogenic amines like beta-alanine, GABA, and serotonin, we already assay 3. Only the beta-alanine shows the significant overexpression in male, but not in female. So, and the carboxylic acid like acotinic acid, hippuric acid, lactic acid. So, only lactic acid shows the overexpression in male, but not the aconitic acid and hippuric acid do not show any significant changes in between two sets. And pyraxyl sulfate is also don't show any changes. And hormone like cortisols also showed significant overexpression in male. And the indole derivatives like 3-IAA indoleacetic acid, the indole propionic acids, indoxy sulfate do not show any significant changes between the males and females. And nucleic acid bases like hypoxanthine, xanthine, and these two compounds, these two nucleic bases have no significant changes, but choline have significant changes in male overexpression than female. And in case of fatty acids, so we only show that fatty acid do not show any significant changes. So I cannot say strongly that this indicate that most of the metabolites are overexpressed in male because it is a less sample size. So after completing the 5,000 sample analysis, we can strongly say the status of the metabolites reference range in Indian populations, males and females. And later we will analyze the data in different diseases like cardiometabolic disorder, cancers, what are the metabolite changes as for because this is a follow-up study. So here normal is that if today I am normal, after two years I may be the cancer the disease. So it will be the actual control to person. So it will be actually the personalized risk prediction course. So this is our team in proteomics and metabolomics research unit is CSIR IGIB Delhi. So we have member Dr. Santanu Sengupta, me, Rajat, Kasid, Mamta, Sibani, and Diksha, and Reibu. Yeah, thank you. So if any questions, I'm open to any questions.